When we speak of the shape of an object, we do not mean a collection of figure elements. We mean by and large a collection of spatial relations between figure elements. In the same way, when we speak of the tune of a song, we do not mean a collection of notes. We mean by and large a list of frequency relations and time relations between notes and fragments of notes. In general, it can be said that to a good first approximation, such things as shape and tune are made up of the results of comparisons between pairs of elements. In this video, I would like to say a few words about the way that a network of neurons can handle such comparisons, such relationships between simple elements of more complex objects. First of all, it is not immediately clear that this is possible at all, because assuming that the simple elements being linked are represented by ignitable neuron groups, it would seem that in order to communicate a relationship, one neuron group would have to make reference to another neuron group and say something about it to a third one. But of course, the language of neurons is not very expressive. All they can do is fire, and all the neuron groups can do is ignite. However, as it happens, that is all they have to do. Two ignitable neuron groups can synchronize their ignitions to one another and thereby broadcast the fact that their messages are related. Now, this, of course, still does not solve the problem, because synchrony between two ignitions can only express that the two underlying elements are related to one another and says nothing of the nature of the relationship. However, this is where the chemical signature of neuron groups enters the picture and supplies the missing piece. Let me illustrate this on a concrete example which involves the direction-coded cell groups that I mentioned in the last video, and show how synchrony between groups of such cells can unambiguously communicate the relative direction of their receptive field locations. By the way, I discuss all of this in much more detail in my book. The idea is that these cells are in the primary visual cortex, V1, but the cells that must read the messages about the relationships are in the next visual area, V2, to which all the direction-coded cells send axons. Let us say that a cell located in V2 detects two sets of volleys, and it recognizes from their labels that they come from two groups of oppositely directed direction-coded cells. I drew this slide in such a way as that the neuron group at the top sends its axon in the 4 o'clock direction, and the one at the bottom in the opposite direction, in the 10 o'clock direction. Let us say that the cell in V2 also detects that the volleys from the two cell groups are consistently in sync with one another, like this, which means that at least one of the cell groups is able to follow the other, meaning that it receives synapses from it. Incidentally, synchrony would usually not imply direct synaptic contact, but in my book I show that it is possible to design the link-up protocol between the groups in such a way that it does. Now, if either of the two groups receives synapses from the other, it means that it is within the field of arborization of the other group's axons. Therefore, the 4 o'clock pointing group is in a 10 o'clock direction from the 10 o'clock pointing group, and vice versa. In other words, the two groups have to be situated like this. Otherwise, the axons of one group would miss the cells of the other. And, thanks to the chemical labels carried by the cell groups, the cell in V2 is in a position to read this information. What this means, looking at it from a different point of view, is that the that two groups of oppositely direction-coded neurons can broadcast their relative direction by synchronizing their firing.